Folate's influence on cognitive longevity represents one of the most quietly promising areas in neuroscience and aging research. While its traditional identity as a vitamin essential for prenatal development and red blood cell formation is well established, folate's role in brain aging is emerging as a powerful, if understated, force in protecting against neurodegeneration and preserving cognitive performance well into older age. The brain, an organ of extraordinary metabolic demand, relies heavily on nutrient support to maintain the integrity of neurons, neurotransmitters, and the microvascular network that sustains them. Folate, through its deep integration into methylation pathways and DNA repair systems, has become a central focus in the effort to understand and prevent age-related cognitive decline, including Alzheimer's disease. At the heart of this connection is folate's involvement in homocysteine regulation, a recurring and critically important theme in neurovascular health. Elevated homocysteine is a well-known risk factor for cognitive impairment and Alzheimer's, not only because it damages blood vessels, but also because it exerts direct neurotoxic effects on brain tissue. High homocysteine promotes oxidative stress, impairs neurotransmitter synthesis, and triggers chronic inflammation, all of which are implicated in the neurodegenerative process. Folate helps lower homocysteine by donating methyl groups that convert it back into methionine, essentially neutralizing this threat before it can erode cognitive capacity. Numerous studies have shown that individuals with higher folate status tend to have lower homocysteine levels and perform better on memory, attention, and executive function tests, especially in aging populations. Beyond homocysteine control, folate contributes directly to the synthesis of critical neurotransmitters such as serotonin, dopamine, and norepinephrine, chemicals that not only influence mood but also cognitive sharpness and emotional regulation. The methylation reactions required for their production depend on folate as a foundational nutrient. When folate is insufficient, the brain's ability to produce and recycle these neurotransmitters falters, leading to subtle but progressive declines in mental clarity, focus, and mood stability. These effects may precede more visible signs of cognitive impairment and may be reversible or at least modifiable with timely nutritional intervention. One of the more intriguing avenues of research explores folate's role in the formation and clearance of amyloid beta plaques, hallmarks of Alzheimer's pathology. While the mechanisms are still being elucidated, animal studies and early human research suggest that folate deficiency may impair the expression of genes involved in amyloid processing and reduce the brain's capacity to eliminate misfolded proteins. Furthermore, methylation defects in folate-deprived states can lead to the dysregulation of genes that control inflammation and neuronal survival, potentially accelerating the onset or severity of Alzheimer's-related changes in the brain. By supporting balanced gene expression and limiting inflammation, folate may act as a silent modulator that delays the epigenetic drift toward neurodegeneration. Cerebral blood flow and vascular integrity, both crucial for cognitive longevity, also appear to be supported by folate. The brain's vast capillary network delivers oxygen and nutrients to neurons, and any disruption in this system, whether through atherosclerosis, microbleeds, or impaired perfusion, can compromise cognition. Folate's role in reducing endothelial dysfunction and oxidative stress may help preserve this network, especially in the hippocampus, the memory center most vulnerable to aging. By protecting the small vessels that feed the brain, folate indirectly safeguards the neurons they support. The question of how much folate is needed for optimal cognitive protection is still under investigation, but emerging data suggest that both dietary intake and blood folate levels should be maintained in the upper normal range, especially in older adults. Importantly, genetic variations in the MTHFR enzyme, which converts folate into its active methylated form, may impair cognitive resilience in certain individuals. These people may benefit more from supplementation with L-methylfolate rather than folic acid, further emphasizing the potential for personalized approaches in cognitive nutrition. What makes folate's cognitive benefits particularly compelling is their subtlety. Unlike drugs that produce noticeable changes in symptoms, folate acts over years or decades to maintain the infrastructure of neural health. It slows the erosion of brain networks, quiets low-grade inflammation, and reinforces the body's ability to correct cellular errors before they accumulate. In this way, folate doesn't simply support short-term brain function. It contributes to the architecture of long-term cognitive preservation. In the grand narrative of aging, where so many interventions arrive too late to reverse damage, folate offers a preventative strategy grounded in biology, not hype. It protects the brain quietly, daily, and without side effects. While it may not yet be widely marketed as a cognitive enhancer, folate remains one of the most promising nutritional tools we have to influence how the mind ages, how memory endures, and how the brain resists the pull of Alzheimer's disease.